what to look for in this video. This one's really quite short. The major thing that you're going to be looking for, going to be interested in is, well, first of all, there's a question that's going to ask you about to identify an RNA versus DNA nucleotide. So if you don't remember how to do that, maybe take a second and review the nucleotide videos from previous instruction. Identify an RNA versus a DNA nucleotide, and then where does a new nucleotide get added? To a growing strand. To a growing strand. That's it for this one. How do we build a strand of nucleic acid when we're talking about DNA or RNA? Just reminding you, these are images that we've seen multiple times before. A nucleotide is the building block of a nucleic acid. Nucleic acids are DNA and RNA. In this image, quick review, pictured on each side is DNA and RNA. Take a moment to decide which is which, and how can you tell which is DNA, which is RNA. Okay, hopefully you remembered and identified that over here we have that oxygen in addition to the hydrogen, so this is a hydroxyl group, and if we have that additional oxygen in comparison to the other nucleic acid, then this is RNA, whereas this one's DNA because it is a deoxygenated deoxyribonucleic acid. I bring this up again, hopefully you're sick of hearing it, you're like we know, because it's going to be important to what we're talking about today when we're talking about how to extend a strand of DNA. The three parts of a nucleotide are the same for both DNA and RNA. I want to remind you of our carbons. These are going to be important, as you know already. The primes, and just point out a couple of things. So if we're looking at our pentose sugar here, and in this particular image, this is ribose sugar, so this is in RNA. Subsequent images will be DNA, because to emphasize the fact that it's the same in both. If we look at our 1' here, our 1' carbon, we can see that our nitrogenous base is attached there. If we look over here at our 5' carbon, we can see that the phosphate group is attached there. Our 4' carbon is then anchoring here. Our 4' is anchoring this carbon, which anchors our phosphate group. 3 out of our 5 carbons are bonded. You can think of them as taken, they're occupied. And then we have these the 3' carbon and the 2' carbon down here at the bottom of this pentose sugar. So these are the only 2 carbons available to bond to when we start adding nucleotides. And maybe just keep in mind, DNA runs 3' to 5' or 5' to 3'. So right there, you know that it's not going to be this one. It's not going to be the 2' sugar. If we're going to add to a strand of DNA, this nucleotide that's up here in the top left, here, we're going to add another nucleotide to that, because that is exactly how you extend a strand of DNA, is by adding individual nucleotides. Those individual nucleotides carry individual bases, and those bases have to base pair with the other side with a complementary strand. what gets added to either DNA or RNA. In these images, I don't have that extra oxygen. This is DNA. What gets added to either of those is not what we've been seeing as a nucleotide already incorporated into DNA, but what a nucleotide that has 3 phosphates. So this is a nucleotide triphosphate. And this nucleotide that's part of DNA is a nucleotide monophosphate. Both nucleotides, but they have 1 phosphate, or do they have 3 phosphates? That's just how they get named. When a nucleotide is getting added, the nucleotide triphosphate that's going to get attached to the growing strand is brought into contact with that strand. And we'll talk about that in a lot more detail. So if you're like, how, why, what, cool, we are going to talk about that. It's brought into contact with the growing strand. And so that top nucleotide is representing our growing strand. Then what happens is that the 3' phosphate group, the 3', not phosphate group, hydroxyl group, that hydroxyl group is the OH, it's charged and it attacks, this is the terminology, it attacks that bond between the phosphates. And we'll talk more about why is it that specific bond. In RNA, why is it the hydroxyl at the 3' rather than the 2'? We'll talk more about that later. This is happening in a very specific orientation. It's going to attack that bond. And that cleaves or cuts those 2 phosphates away and provides the energy. When those 2 phosphates get cut off, it provides the energy to attach the remaining phosphate to the 3' carbon of the nucleotide that's already part of the chain. So you may remember this from physiology, talking about adenine triphosphate and how its cellular energy, cutting it up is what provides cellular energy. So it provides the energy to bond the first phosphate to the growing chain. And we're going to talk about this in more detail, but you need to know this up front because understanding the mechanism will help you understand the entire process and help you remember that nucleotides can only be added to the 3' end of a growing strand. Only. Nucleotides can only be added to the 3' end of a growing strand. Remember that I pointed out over here at the 5', we've already got a phosphate. There's already a phosphate attached there. It can't be added. We can't add another nucleotide there. No, we can't add a nucleotide there. We can only add a nucleotide where that free hydroxyl group is available to attack that bond and provide the energy to bond the phosphate to the growing chain. So a little preview of what's coming up. The big take home here is only be added to the 3' end of a growing strand. And that is going to be a big theme that's going to be really important to remember over the coming weeks.